Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about this metabolic pathway called glycogen synthesis. As we mentioned in previous videos, humans have three metabolic states: the postprandial, what happens after a meal, early starvation, and the late starvation state. As we saw in earlier videos, after a meal, your insulin levels are high, and the insulin stimulates a lot of synthesis, protein synthesis, fatty acid synthesis, as well as glycogen synthesis. So glycogen synthesis is called glycogenesis, and glycogenesis happens mostly in the liver and the muscles because that's where most of your glycogen is stored. So today we shall focus on this metabolic pathway that becomes very active after a meal called glycogenesis. So before we talk about glycogenesis, just a little bit of background material. The saliva as well as your pancreatic juice contains an enzyme called amylase, and we'll talk about this enzyme because it's clinically very important for the diagnosis of pancreatitis. We'll talk more about this later on. But this enzyme basically breaks polysaccharides such as your starch and your glycogen into a disaccharide called maltose. In the small intestine, we have these enzymes that are located on the surface of enterocytes. These enzymes are called disaccharidases, and the disaccharidase called maltase will break down the maltose into glucose. Other disaccharidases are also present on the surface of the enterocytes. So for example, isomaltase will break down isomaltose into glucose, 2-glucose. Sucrase will break down sucrose into glucose and fructose, and the enzyme lactase will break down the disaccharide lactose into its components, glucose and galactose. So once the glucose is formed, in the next step, we have this transport protein called SGLT, sodium-dependent glucose transporter, SGLT. And this transport protein basically brings glucose into the cell together with sodium. Okay, so it needs the help of sodium to get the glucose into the cell. This protein is therefore a symporter because it brings two different chemicals in the same direction, in this case from the GI cavity into the enterocyte cytoplasm. Now there is a sodium gradient present across this plasma membrane. So it's a high sodium in the GI cavity and a low sodium concentration in the cytoplasm. So this SGLT uses this sodium gradient to help bring in the glucose from the GI cavity into the cytoplasm. What keeps the sodium concentration in the cytoplasm low is this protein here called the sodium-potassium ATPase, the sodium-potassium pump. This transport protein continuously transports 3 sodium out of the cell in exchange for 2 potassium coming into the cell. So basically, sodium is being continuously transported out of the cell. Of course, this pump uses ATP energy to do that. So this pump maintains a low sodium concentration inside the cytoplasm, thereby maintaining this sodium gradient. So this glucose that's present in the cytoplasm is then transported out of the cell into the tissue space or the extracellular matrix, and that is done by this protein called GLUT2. So this is an example of carrier-mediated transport. So in this case, the glucose concentration, which is very high in the cell, is being transported to the tissue space where the glucose concentration is very low. It's important to note that transport proteins such as GLUT2 and SGLT, just like enzymes, they can become saturated. So for example, you can see in this plot, the rate of glucose transport increases with increasing glucose concentration, and once the glucose concentration reaches very, very high, then the rate of glucose transport reaches a maximum, a Vmax, very similar to enzyme kinetics. So the glucose inside the tissue space, the extracellular matrix, will now enter the bloodstream through pores that are present between the endothelial cells that line the capillaries. So this kind of transport between cells is called paracellular transport. So the glucose enters from the tissue space into the bloodstream via paracellular transport. Note that the glucose present in the GI cavity cannot enter the matrix or the tissue space by paracellular transport because there is this tight junction present between one enterocyte and the enterocyte next to it. So this tight junction prevents paracellular transport of glucose. So due to this tight junction, the enterocyte is divided into two parts. You can see this part here would be the apical part, and then we have this part here that is the basolateral part. Okay, so the apical part of the enterocyte is quite different from the basolateral part. So this is how glucose in the GI cavity enters the tissue space and finally enters the bloodstream. And once in the bloodstream, this dietary glucose will now go to the liver through the portal vein. A quick reminder about glycogen structure that we talked about in a much earlier video. Now, glycogen is found in all cells in a form of granules, glycogen granules, which are about less than 40 nanometer. These glycogen granules contain glycogen and the protein glycogenin, plus all the enzymes necessary for glycogen breakdown as well as glycogen synthesis. Now recall that glycogen is a very, very highly branched structure. It has the alpha-1,4 glycosidic linkages, and wherever the branches are, there is the alpha-1,6. Okay, so it's all alpha-1,4, and whenever there's a branch point, there will be an alpha-1,6. Okay, otherwise it's all alpha-1,4, and again, wherever there's a branch point, there's alpha-1,6. So that's a basic structure of glycogen. Because of this structure of glycogen, it has only one reducing end, and that reducing end is actually connected to the protein called glycogenin. The rest of the terminals of glycogen at the end of every branch, these are all non-reducing ends. Okay, so glycogen has many non-reducing ends. And that's just important because you'll see glycogen synthesis as well as glycogen degradation occurs at these points here. Okay, with that background, let's look at the key steps in glycogen synthesis. We'll focus on the liver and muscle because that's where most of the glycogen metabolism occurs. In case of liver cells, the glucose in the bloodstream is brought into the liver cell by this transport protein called GLUT2, while in case of muscle cells, there is another protein called GLUT4 which brings in the glucose into the muscle cells. Now GLUT2, this protein here, has a very low affinity for glucose. It has a very high Km, 20 millimolar. Remember, the blood glucose concentration is only around 5 millimolar. So only if the blood glucose concentration becomes very high, and you only see that postprandial after a meal. So only when the blood glucose concentration becomes high will we see an appreciable amount of glucose entering the liver cell. Now GLUT4, which is found in the muscle cells as well as adipocytes, GLUT4 is found inside the cytoplasm during fasting conditions. Only after a meal will this GLUT4 travel from the cytoplasm to the plasma membrane, and only once it is in the plasma membrane, then glucose can be transported into the cytoplasm. Okay, so again, postprandial is the only time when the insulin levels are high. Insulin will cause this GLUT4 to travel from the cytoplasm to the plasma membrane and thereby bring in glucose into the cell. So this is one of the major functions of insulin. Insulin levels are high after a meal, and insulin, one of the effects of insulin is that it brings glucose from the bloodstream into cells. In other words, insulin tends to decrease your blood glucose concentration. So glycogen synthesis begins from this protein called glycogenin. So glycogenin, we see the tyrosine hydroxyl group of glycogenin, and to this hydroxyl group will be connected 8 glucose units. These glucose come from a reactive form of glucose called UDPG, which we'll talk about in a few seconds. So attachment of this 8 glucose to glycogenin, 8 glucose attached to glycogenin, this gives us what we call a glycogen primer. So this glycogen primer can then be extended. More glucose units could be attached here, branch points could be made here, and then more glucose units attached to these points here, and so on, making the glycogen longer and longer with more branches. So let's see the various steps by which this glycogen primer is made into glycogen. So the glucose that is brought into the cell reacts with ATP, and that will give us glucose 6-phosphate and ADP. So we get the glucose that is brought into the cell is converted into glucose 6-phosphate. This is an irreversible reaction, and glucose 6-phosphate is a very important intermediate, not only in glycogen metabolism, but we saw glucose 6-phosphate in glycolysis. We will see that in another pathway called hexose monophosphate shunt, and we'll also see that glucose 6-phosphate is an intermediate of gluconeogenesis in future videos. Now in the liver, the enzyme that catalyzes this reaction is glucokinase or GCK, and in the muscles and other cells, it's the enzyme called hexokinase. So just like the transport protein GLUT2, glucokinase also has a very low affinity for glucose. The Km is about 10 millimolar, that's about 180 milligrams per deciliter. Hexokinase has a much higher affinity. The Km is only about 0.1 millimolar. So although hexokinase has a higher affinity for glucose than glucokinase, the problem with hexokinase is that it can become saturated very fast. So it cannot handle large amounts of glucose. However, glucokinase doesn't become saturated easily, and that will cause a lot of glucose to be phosphorylated into glucose 6-phosphate without the enzyme becoming saturated. So between the glucose transporter GLUT2 in the liver and glucokinase in the liver, large amounts of glucose can be brought into the liver cell and converted into glucose 6-phosphate. Now, insulin, which increases after a meal, stimulates the activity of glucokinase as well as the activity of a isoform of hexokinase called HK2. In the next step, the glucose 6-phosphate is converted to glucose 1-phosphate. This is a reversible reaction catalyzed by the enzyme phosphoglucomutase. The glucose 1-phosphate is now made into a reactive form of glucose called UDP glucose. So basically, glucose 1-phosphate will react with a nucleotide. In case of humans, the nucleotide is UTP, not ATP, UTP, forming this very reactive form of glucose called UDP glucose or UDPG. So although this part of the reaction is reversible, we have a product pyrophosphate form, and pyrophosphate is immediately broken down to 2-phosphate by the enzyme inorganic pyrophosphatase that is present in all cells. So overall, this reaction, combine this and this reaction where glucose 1-phosphate reacts with UTP to give you the activated form of glucose UDPG and the 2-phosphates, this reaction is irreversible. So the conversion of glucose 1-phosphate to UDPG glucose, UDP glucose, is overall an irreversible reaction. It can only go in this direction. Note that this enzyme, UDP glucose pyrophosphorylase, is named after the reverse reaction, where the UDP glucose reacts with PPI to give you UTP and glucose 1-phosphate. So the attachment of a UDP to carbon number 1 to give us UDP glucose makes the glucose very reactive. The hydroxyl group at carbon number 4 of the glycogen chain can now react with carbon 1, and that will kick out the UDP, which comes out as a leaving group. So in organic chemistry, the UDP acts as a leaving group, and that will lead to carbon 4 joining carbon 1, forming an alpha-1,4 glycosidic bond. So in this way, we can add glucose continuously to the non-reducing, and again we can add another glucose. This can be repeated to form another glucose, add another glucose, and so on, thereby extending the chain, making alpha-1,4, 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 and making the glycogen chain longer and longer. So important to note that this extension of the chain happens at the non-reducing ends. And since glycogen has many non-reducing ends, so we can extend the glycogen at many, many different parts simultaneously. So this enzyme that catalyzes this reaction, where glucose is being donated to a glycogen to increase the length of the glycogen, this enzyme is called glycogen synthase, and this reaction is irreversible. Now, glycogen synthase is a very important enzyme that controls how fast glycogen is made. This enzyme is found in 2 forms. We have the phosphorylated form of glycogen synthase, which is called GSB, This form is not active. It doesn't catalyze the reaction. Then we have the dephosphorylated form, the one that doesn't have phosphates attached. This is called GSA. A here stands for the active. This form is the active form. This is the form that will catalyze this reaction. There is another enzyme called glycogen synthase kinase 3, GS kinase 3. This is the enzyme that causes the phosphorylation of the GSA. Remember, the A is the unphosphorylated form. So the A, which is the active unphosphorylated form, when it gets phosphorylated by ATP, then it becomes the inactive B form. Now insulin, which is released after a meal, it will cause the inactivation of GS kinase 3. And if this enzyme becomes inactive, then this reaction will not happen, and now we have glycogen synthase in the A form. Glycogen synthase in the A form is active, and that will increase glycogen synthesis. So insulin will increase glycogen synthesis, which is what we see after a meal. Okay, so in the postprandial state, insulin levels are high, and insulin will stimulate glycogen synthesis. So glycogen synthase makes the alpha-1,4, but what about the branches alpha-1,6? How do we form a branch? That is done by an enzyme called branching enzyme. It has a long name, amylo-1,4,1,6 transglycosylase. Basically, what this enzyme does, it takes a 7-residue glycogen chain from a terminal and then adds it to a C6 hydroxyl group on another part of another chain or the same chain. And that will lead to the alpha-1,6 branch that is formed. So this is a branching point. And that's how the branches of glycogens are formed, by the enzyme called branching enzyme. So everywhere where you see a branch, that will be alpha-1,6. Okay, so let us summarize the synthesis of this very, very important fuel. This very important fuel, especially liver glycogen, although the muscle has its glycogen too, but liver glycogen, as we have seen in previous videos, is what maintains your blood glucose during the fasting period. During times when you are asleep or between meals, this is the glycogen that maintains our blood glucose. And maintaining the blood glucose, as we mentioned earlier, is very critical for red blood cells and for the brain. So let's summarize this very important pathway. So the enterocyte in the small intestine is the cell that will absorb glucose into the body. In this enterocyte, the concentration of sodium inside the cytoplasm is always kept very, very low. And that is done by the sodium-potassium pump that continuously pumps sodium out using ATP energy. So since sodium inside the cell is very low, sodium outside the cell in the GI cavity is very high. This sodium can come from your diet or it can come from the pancreatic juice. So this sodium that is much higher on the outside in the GI cavity compared to the inside of the enterocyte, this sodium will continuously enter because of the sodium gradient. Remember, the sodium gradient is a form of energy, and that energy really was made by this sodium-potassium pump. So sodium keeps on coming into the enterocyte. Now, since this SGLT protein not only brings in sodium, it simultaneously also brings in glucose. So in this respect, using the sodium gradient, glucose from the GI cavity will enter into the enterocyte. As more and more glucose enters the enterocyte, there's a possibility that the concentration of glucose is very high inside the cell, while the concentration of glucose might be very low in the GI cavity. But this does not matter, because although the concentration is lower in the GI cavity, all the glucose will still keep on coming into the cell. Why? Because of this driving force by the sodium gradient, because the sodium is continuously entering the cell, and that drives all the glucose in the GI cavity to also enter the cell. So suppose we had a protein like this GLUT2. Here, suppose we had that protein on the apical membrane, we would run into a problem, because once the glucose concentration here is low and the glucose concentration here is high, the glucose transport would stop. Okay, but in this case, SGLT, it doesn't happen, and this protein makes sure that all the glucose in the GI cavity will be absorbed into the enterocytes. Once in the enterocyte cytoplasm, then GLUT2, which is found on the basolateral membrane, GLUT2 will transport this glucose out into the tissue space, and from the tissue space, the glucose will enter the capillaries, and from this blood vessel, the portal vein, this glucose will now go to the bloodstream of the liver. So now the glucose in the bloodstream of the liver will be taken into the liver cell, the hepatocyte, by this protein GLUT2, and the glucose will be converted into UDP glucose. So by converting glucose into UDP glucose, this carbon 1 here of the glucose becomes reactive by attaching a UDP group to carbon 1. So now this hydroxyl group at position number 4 of the glycogen primer will be able to react with carbon 1, and this will cause the attachment of a glucose to this carbon 4. So carbon 1 gets attached to carbon 4 by a alpha-1,4 glycosidic linkage, and that will cause the formation of a new carbon 4 hydroxyl, which can repeat this process, react with another UDP glucose and add another glucose unit here to alpha-1,4 and another alpha-1,4 and so on. So the glycogen chain can be extended by adding more and more glucose to every branch. So while glycogen synthase makes the alpha-1,4 and extends the chains, the branching enzyme makes alpha-1,6 to make branches on the glycogen. And then these branches again can be extended by the glycogen synthase reaction. So in this way, glycogen in the liver that was used for maintaining blood glucose during the early starvation period, like for example when you were asleep, all this glycogen that was used up is remade, is replenished, and that happens after a meal. In the next video, we'll talk about the synthesis of another very important fuel that happens after a meal, and that is fatty acids. Okay, so we'll talk about the synthesis of fatty acids, which is the second most important fuel after glucose, in the next video. So until then, once again, thank you very much for watching these videos. Goodbye and have a healthy and happy day.